I don't know if you got my announcement, but we're just doing the Q&A session today because I don't have anything new to add. So just anything that you want me to clarify from those videos, I'm here to do so. Anyone, anything?
Yeah, I don't have any questions. I was just coming in to listen.
Okay.
I know you have an exam review that's online, but if you could verbally just give a quick overview of everything, I think that would help kind of stick.
All right. So some things that I think you should be focusing on. Actually, hold on. Where did I put my book? Here. I'm just gonna go along with the textbook. And see where we are. Okay, so from the lymphatic system, chapter 21, I think that one is it's pretty straightforward. So make sure that you know what is what, how things are organized, how those organs are organized. Make sure you know the difference between primary and secondary structures, be able to classify them, and then also why we classify them that way. So like your primary lymphatic structures are responsible for formation and maturation. Secondary are going to house lymphocytes. That's kind of like your basic division. And then lymphatic organs versus lymphatic nodules. Organs would have a complete capsule. Nodules are not enclosed in a structure with a capsule. And then make sure that you know which ones are which, right? So primary structures are red bone marrow and we have some more people coming in. So your primary structure, primary structures are your red bone marrow and thymus, and then secondary structures are everything else. Just know the basic location and terminology that goes with those things. And that's pretty much it for chapter 21. 22, the introduction, I think is pretty simple to read. Be aware of the things that can cause infection or they're harmful to our body. For your locations of the immune cells, again, make sure which ones are located, which ones are located where. So because if you look, like it's going over, these are the locations where you can find these cells, these immune cells, but different locations have different preferred cells that we can find there. So like you can see in the skin and mucosal membranes, you have dendritic cells, and that's because of how the cells of the skin and mucosal membranes are arranged, right? Those dendritic cells can extend their dendrites through those tightly packed cells of those tissues. So that's for that. But I think each section there, it tells you epithelial layers, you see the dendritic cells, connective tissue, you see mostly mast cells, things like that. For be aware of what cytokines are. They're simply chemicals that are released by immune cells. And there are some examples of cytokines. I'm not going to go into details about those, but just be aware that interleukins, which you see at the end or towards the end of chapter 22, or interferons, which are part of the innate immunity, those are types of cytokines. And then there are some other ones. Tumor necrosis factor, we really don't talk about much more other than in that table, but I think its name pretty well suggests what is it. So tumor necrosis factor would be killing tumor cells. And then colony stimulating factors, you've already seen those in chapter 18 when we talked about development of formed elements, right? So colony stimulating factors are those chemicals that are going to affect the development of formed elements. And that's all you really need to know about that. When you, I don't know if you're looking at your textbook here, but there's figure 22.2 that divides the immune system for you into the innate and adaptive immunity. And that's a very important table. You should make sure that you have that big picture for the immune system using that table there. So you have the innate immunity, you have the adaptive immunity. Your innate immunity, you have there are actually two lines of defense there. There's a first line of defense, which would be your skin and mucosal membranes. That's your entry prevention. Okay, so that's your first line of defense, and it's part of innate immunity. If that fails, innate immunity is going to still continue. We have internal processes for the innate immunity, and that's going to be your cellular response. So different cells are going to do certain things if bacteria or viruses are allowed to enter our body. You're going to see chemical response with the interferons, and then you also are going to see physiological responses such as fever and inflammation. Okay, so these processes are going to be non-specific, and so innate immunity is non-specific. It doesn't really care what kind of bacteria it is or what kind of virus it is going to work pretty much the same way, no matter what the pathogen is. Then as a result of this innate immunity, and this is especially visible when you talk about or when you look at the inflammation, you can see how the process of inflammation, it produces this exudate and traces of the pathogen are going to enter lymphatic system. And once that happens, this is what activates your adaptive immunity. And so for the adaptive immunity, when we talk about that, you have to be able to focus at the T lymphocytes and B lymphocytes and what they do. Be aware that B lymphocytes give rise to plasma cells ultimately. The action of the T lymphocytes and B lymphocytes as part of adaptive immunity is what's going to produce a specific response. So this part is going to be specifically designed against a particular virus. So not just the virus in general, but are you a flu type A virus? Then it's going to be producing antibodies and specific response against that flu type A, not against type B, but against type A. So that's how specific that is. Whereas with the innate immunity, there are some cells that prefer bacteria, for example, neutrophils. Okay, they prefer bacteria, but they don't really care what kind of bacteria it is. So it's non-specific for the innate part. So be able to talk about the 3 lines of defense, right? Your first line of defense is preventing entry. Second line of defense is the non-specific internal defenses. And for that second line, you see the cells, the action of individual cells. You see the action of chemicals like interferon, and complement system also, I haven't mentioned that. And then you see those physiologic responses. That's your second line of defense. And then third line of defense is your adaptive immunity. Okay, so if you look, I think it's on after the innate immunity, there is that integrate, the 2-page thing. So it shows you first line of defense, and it actually lists on that page with the little baby with the toy. It actually lists more of the preventing entry, how our body prevents entry and gets rid of pathogens that are trying to enter our body. And then you see the second line of defense next to it. And then your adaptive would be third line of defense there. When we look specifically at adaptive immunity here, this one is a little more intense, I think, than the innate immunity. So here, some terminology that might not be something that you would understand right away is the effector response. It's just kind of a weird way that they're wording that. Maybe you have not heard that before, but effector response is what is that cell type ultimately doing? So effector response for your T lymphocytes, this is going to depend on what T lymphocyte we're talking about. So for the cytotoxic T lymphocytes, effector response is killing of the infected cell. And effector response for the helper T lymphocyte is activation of other immune cells and other lymphocytes. Effector response of B lymphocyte is going to be production of antibodies, and it's basically the action of the antibodies here. We call the T lymphocytes are associated with cellular or cell-mediated immunity. The B lymphocytes are associated with humoral immunity. Okay, humoral refers to the plasma of your blood. And it's called humoral because the effector response of B lymphocytes is the production of antibodies. And you find the antibodies in the blood plasma. So associate the humoral with blood plasma. Okay, don't forget that. The action of antigen and antibody, for that section when it talks about antigens, make sure that you are familiar with the terminology, with the vocabulary. So what is antigenic determinant? What is epitope? What is immunogen? Things like that. That's just vocabulary. You've got to memorize those things. So I'm gonna let you do that. Briefly know how the autoimmune diseases work, which, you know, that's a huge, big thing. And they're giving you what, like half of a little paragraph about autoimmune diseases, but this is basically where your immune system fails to recognize own antigens as self-antigens, and it starts attacking your own cells. And this can happen because you either you are exposed to antigen that is very similar to your self-antigen, or your self-antigen mutated, at which point it is no longer a self-antigen. And so whenever you treat autoimmune disorders, usually treated by things that are going to suppress your immune system quite a bit. So that's that. And I don't know, I forgot if I said that in the recording, but if you have seen Little Women, the movie, whichever one, or you have read the book Little Women, one of the sisters, I think she might be the youngest one, I don't know, but she's ill, and this is because she, as a child, has had strep that developed into scarlet fever, and this often results in damage to the heart, because what happens is the antigens that cause that scarlet fever are very similar to self-antigens on the heart. And your immune system basically starts attacking the heart. And that's what was wrong with her in Little Women. If you haven't seen it, watch it. It's a good movie, I think. It's a classic. So, or read it. So anyway, so that's the autoimmune diseases. General structure of lymphocytes, be aware what these lymphocytes look like, that they have receptors. Your T lymphocytes also have co-receptors. And you do need to know that your helper T lymphocytes have CD4 co-receptors, your cytotoxic T lymphocytes have CD8 co-receptors, and then the receptors on the B cells look like antibodies. So they literally are antibodies, okay, at that point. Another important topic or concept is that the T lymphocytes, just both classes, cannot really recognize an antigen, like a foreign antigen, on their own. They have to have another cell present that antigen to these T lymphocytes. And we call these cells antigen presenting cells. So write it down and make sure that you are able to tell me the 3 possible antigen presenting cells. And these are your B lymphocytes. So B lymphocytes can act as antigen presenting cells, macrophages, and dendritic cells. Okay, so your 3 antigen presenting cells are B lymphocytes, macrophages, and dendritic cells. So, and these are the cells that will process the pathogen. They will destroy it in the process of this, and then they will express this antigen on their surface. And that's where T lymphocytes is like, oh, this is what I need to, start fighting and attacking. So it's kind of an important part of activation of those T lymphocytes. For the MHC molecules, when you look at figure 2211, you can see what's happening in a healthy cell. So your normal healthy cells are producing, part of their function is that they're producing this protein that's called MHC molecule. And this MHC molecule is like a holder for an antigen. And think about your self antigen as an ID tag. So this lets your immune system cells know that this cell belongs here and they should not attack it. Okay. If you don't have that self antigen, or that self antigen is different, then immune system cell knows that they need to attack that cell. Okay. So in a healthy cell, you see the MHC molecule produced, you get this self-antigen molecule that's synthesized in the cell attached to it, and then it goes to the cell membrane and basically becomes a cell membrane, part of the cell membrane, with the self-antigen sticking to the outside. So it's like a marker telling your body, I'm okay, don't kill me. Okay, that's your self-antigen. The self-antigens are going to be attached to MHC class 1 molecule, okay? Always, okay? You can have MHC class 1 molecule carry a foreign antigen as well. So this is if this normal cell that minds its own business, let's say this cell gets attacked by a virus or something. So in this case, in the example, if you're looking at the picture, the cell gets attacked by a virus. This cell, once it's infected, we don't heal cells. If a cell gets infected and damaged, your body needs to go and destroy it. So if the cell gets infected, what you're going to see is it's going to try to destroy the virus or the pathogen. And as it's doing that, the pieces of this pathogen will take place of that self-antigen. So now you have MHC class 1 molecule with a piece of the virus or bacteria or whatever. Okay, you have a MHC class 1 molecule with the foreign antigen. So now that says to your body, this cell has been infected and it needs to be destroyed. Out of the 3 lymphocytes that you have T lymphocytes, the cytotoxic and helper, and then you have B lymphocytes. Who is going to attack this cell? The cytotoxic T lymphocyte, okay? Because this is a cell that needs to be destroyed. Cytotoxic T lymphocyte, it says it in its name, cyto for cell, toxic. And so it's going to, cytotoxic T lymphocyte will ultimately destroy a damaged or unhealthy or infected cell. So what you need to know is that cytotoxic T lymphocytes have CD8 co-receptors. And I think in my video, I say how I remember that cytotoxic is CD8 and helper is CD4. Cytotoxic has 2 O's in the word. When you put 2 O's next to each other and you turn it sideways, it's an 8, right? It's a little stretch, but it works for me. And then helper, a lowercase h, when you turn that upside down, it looks like a 4. So helper T lymphocytes have CD4 co-receptors. So anyway, so cytotoxic T lymphocytes will interact with MHC class 1 molecule with a foreign antigen. Okay, so cytotoxic, MHC1, and they have CD8 co-receptors. Okay, those 3 things go together. Helper T lymphocytes have CD4 co-receptor, and these are going to be the ones that interact with MHC class 2 molecule. So make sure, if you don't have that written down somewhere yet, make sure that you write that down, okay? Cytotoxic, CD8, they interact with MHC1. Helper, CD4, interact with MHC2 molecule. So we have this other MHC molecule, which is the MHC class 2 molecule. And this one is only present on antigen presenting cells. Okay, so you don't find MHC class 2 molecule on any other cells. You only find it on macrophages, dendritic cells, and B lymphocytes. The process is relatively the same whenever this cell gets the hold of the virus or the bacteria. There's one big difference between the two, okay, from what we saw with the infected cell versus MHC class 2 molecule on the antigen presenting cell. When antigen presenting cell engulfs the pathogen, it is not getting infected, okay? It's doing that to destroy the pathogen it breaks it down and then uses MHC class 2 molecule to show the pathogen to the T lymphocyte. And so antigen presenting cells, when they're using MHC class 2 molecule, they're not infected. Now, that's not saying that antigen presenting cells cannot get infected. They can, but we're not really talking about that here, okay? So when the antigen presenting cell presents the antigen, that cell itself is not infected. It's just doing a service to T lymphocytes saying, here, see this, and this is what we need to fight. And so since this cell is not infected, it doesn't need to be destroyed. This is what's going to interact with those helper T lymphocytes. Okay, helper T lymphocytes will then recognize this antigen and get activated and help with activation of other cells there. Okay, so make sure you know which T lymphocytes go with which MHC molecule and which co-receptor, and then know that big difference between, you know, when a cell is infected versus when a cell is just presenting the antigen. Okay, so that's that, because I know that section can get a little tricky sometimes. So there's a little clinical view that you might want to read if you're interested in this, and it talks about the MHC molecules and organ transplants. So you probably heard that, you know, you can't just give an organ to anybody. You have to be a match, right, for certain things. So it's a very short little box that you might want to read and kind of see how that works. But I'm not going to test you on it, okay? It's just something that I think is interesting for you to read. Okay, the next section talks about formation and selection of lymphocytes. So we know that lymphocytes are formed in the red bone marrow. This is where all the formed elements are formed. And then the T lymphocytes are going to travel to thymus. So they get formed as like thymocytes, I think is what we call it, yeah. And when they're formed, when these T lymphocytes, they're not T lymphocytes yet, when these thymocytes are formed in the red bone marrow, they're actually formed with both co-receptors. So they have both CD4 and CD8 co-receptor. And here I am trying to think about what I said about this on the video, but so when these thymocytes enter the thymus, they're going to encounter these cells that are expressing antigens that are not self-antigens. They're like practice foreign antigens. And this is to see, is this thymocyte able to recognize something that's not a self-antigen? Because that's kind of the ultimate goal of that cell. It's supposed to be able to react to a foreign thing, right? And so these thymocytes are going from cell to cell and then, you know, trying to make a match with some of those practice antigens. And if they make a match with MHC class 1 molecule, they will keep their CD8 co-receptor, because MHC1 and CD8 go together, and they will become cytotoxic T lymphocytes. If they make a match with a MHC2 molecule, with an antigen on the MHC2 molecule, this is going to be done using that CD4 co-receptor. They will keep the CD4 and they will lose the CD8, and they will become the helper T lymphocytes. So I think it's not really shown here that it's really the first step that determines this. They're putting that selective loss of CD4 or CD8 towards the end, but it really is that initial match that is going to make that decision whether that cell is going to be cytotoxic or helper. So in that first step, you see binds MHC molecule. If it does, then it survives because it is able to recognize something that's not a self-antigen. So it's making that match there. If it does not make a match with anything, you don't need it. Okay, you're going to kill it. And about 95% of all thymocytes that enter are going to fail this first step. So only 5% of your thymocytes that enter thymus are able to make a match with something. The rest of them are going to die by apoptosis. So this first step is called positive selection. And then they are going to, and this is happening in the cortex of the thymic lobule. As the cells are, passing through the cortex, if they pass the positive selection, they're going to move closer to the medulla. It's really, this is going to be kind of on that junction of the cortex and medulla. And this is where they will encounter cells that are showing self-antigen. Okay, so now these cells are saying, okay, this is the self-antigen. Are you going to attack me? If these cells do make a match with that self-antigen, you have to get rid of them, because this means that they would be attacking your own cells. More than 2/3 of the cells going into the negative selection will fail this step. Okay. And so for here on that table, if you're looking at that figure, this is figure 2215. So if you don't have, and I am recording this, so if you don't have the textbook right now, if you want to kind of go back and refer to the figure as I'm talking about it, you can. So figure 2215, and under the negative selection, it says recognizes self-antigen. And this, I think, is a poor choice of words here because it's not really obviously clear what that means. So I want you to replace the word recognizes with the word binds, just like it was done in the first step. Okay, so that you know that that's what it means, because some people are thinking about that phrase as, well, it recognizes it's a self-antigen, so it's not going to bind. Then we have another half of people that are like, no, it recognizes the antigen, it means it does bind. Okay, so just replace that word recognizes with the word binds. So if the cell is binding to self-antigen, this would be activating that thymocyte, so you don't want that. Okay, you gotta kill them. So ultimately, about 2% of these cells will survive the selection. It's very, very strict, very picky, but it is important. You want to have cells that are able to recognize and fight a foreign antigen, but you also don't want to have cells that are killing your own cells. Okay, so that's that for that selection here. For your activation, so if you're looking at the activation, this is figure 2216. Notice that helper T lymphocyte is kind of in the center of that. This is spread over 2 pages here. And we're going to begin with the helper T lymphocyte kind of in a general. And I want you to understand one thing. These cells are going to be near each other. This is happening in the same area. So it's not like you have an area that only has helper T lymphocytes and another area only has B lymphocytes. They're all doing these things kind of at the same time, and they're co-stimulating each other at the same time. So you're going to see helper T lymphocyte ultimately makes a match with an antigen presenting cell. When an antigen presenting cell shows this antigen to the helper T lymphocyte gets activated and it releases interleukin 2. This interleukin 2 will stimulate that original helper T lymphocyte. That helper T lymphocyte will clone itself, and all the clones of that helper T lymphocyte now are going to have a memory of that foreign antigen. So even though they might not have actually physically made a match with the antigen presenting cell, they're going to be now activated like if they did. So you have a bunch of these helper T lymphocytes that are now producing more and more interleukin and stimulating other immune cells in the area. One of the other cells will be cytotoxic T lymphocyte. Cytotoxic T lymphocyte will make a match with a foreign antigen on the infected cell. And then the interleukin is going to stimulate this cytotoxic T lymphocyte to clone itself. So cytotoxic T lymphocyte will kill that infected cell, but also it now is cloning itself so that more infected cells can get destroyed by these activated cytotoxic T lymphocytes. For both helper and cytotoxic T lymphocytes, group of these clones will become your memory T lymphocytes, and these will be ready if your body gets exposed to that same exact pathogen again. And your second response, because you now have an army, thousands and thousands of these lymphocytes that already know that they need to attack this. Your second immune response is generally so fast, you don't even know that you have been exposed. You don't even know that you were infected because all those cells will just destroy that pathogen before it even has a chance to say hello. Okay. So that's your T lymphocytes. Now B lymphocyte, this is, so if you're looking at the helper T lymphocyte table, we say that antigen presenting cell is activating the helper T lymphocyte. With B lymphocytes are antigen presenting cells. So if the antigen presenting cell for the helper T lymphocyte happens to be B lymphocyte, the sequence of events is a little different here. It involves interleukin-4, okay? And that's going to activate your B lymphocyte, which will differentiate into a group of plasma cells, which are your antibody factories, and then a group of memory B lymphocytes. Again, these will be left behind and stand on alert if you get exposed to that same pathogen again. So those are the most important things about that activation. Just be aware that there are, these things are happening simultaneously, and these cells are activating kind of each other there. So all the cells are necessary, and they're going to be in the same area. Make sure you know how to label antibody. Okay, so that's just labeling all those different regions. Know that for actions of antibodies, it's 3 and 3 when you're looking at that table. Table 22.5, make sure that you know what each action means. So you have 3 actions that are due to the antigen binding site, and then 3 actions that go with the FC region, with the stem part of the antibody. Know which 3 goes with which side, and then know what those words mean. Okay, that's really kind of just definitions. So you will have to spend some time with it. Okay, it's not just going to take you one time me listening to it. You actually have to memorize those things. So know what opsonization means, know what complement fixation means, and that will actually, for the FC region, that kind of takes you back to innate immunity when we first talked about complement systems. So make sure that you know what complement system is. And so that's that. For classes of immunoglobulins, I'd like you to know, I'm not going to test you on the structures of them, but I'd like you to know their unique characteristics. I think that's easier to remember than remembering or memorizing the actions, because the unique characteristics are just easier than memorizing neutralization, agglutination, precipitation, blah, 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 okay? So like for IgG, that's the antibody that crosses the placenta, and so on, okay? But you don't need to know like their half-life or any of that. Locations, primary locations is kind of a good one as well, okay? So primary locations and unique characteristics for that. The last part of 22 would be immunologic memory. So make sure you know what is the primary response, and then subsequent or secondary response. Lag or latent phase, okay, it's that time before your body gets activated. You have that lag or latent phase in the primary response because you're trying to activate the adaptive immunity, okay? And so that takes some time for your cells to realize, there's something going on here. You have to be able to find the leukocyte that is going to recognize that antigen as well, because in primary response, you don't have that army of thousands and thousands of cells that are able to recognize that antigen. You only have a few lymphocytes that are able to recognize that antigen. So that's now we do have a lag or latent phase in the secondary response, but it's much shorter. And that's that. And then for figure 2222, this is dividing active and passive immunity. For active, no, the main difference here is active immunity, your body is producing the antibodies. Passive immunity, you're getting antibodies from another individual. Okay, that's the main difference between the two. Now, both can be naturally acquired or artificially acquired. With your active immunity, your naturally acquired active immunity, this is when you get sick. This is when you get infected by the actual full-blown pathogen and your body has to fight it off. It is producing antibodies as a response to the infection. The artificially acquired active immunity is from a vaccine. And here the idea is that you present or introduce into the body either a weakened pathogen or just fragments of that pathogen. And then your body still kind of recognizes that, like, hey, this is not supposed to be here. And your body is producing the antibodies as a response to the introduction of this weakened or fragmented pathogen. With passive immunity, your active passive immune, or not active passive, your naturally acquired passive immunity would be, this is involving mother and a baby. So this is antibodies from the mom being passed on to the baby, either through the placenta before birth or through breast milk after birth. The artificially acquired passive immunity is from a vaccine. Or not, well, kind of. This is when they transfer the serum. We see the shots against tetanus and botulism being done like this. Also, anti-venom for snake bites is artificially acquired passive immunity. So that's, make sure you know examples of these. I will make you match instances with the type of immunity. Okay, so know that. And then for the hypersensitivities, I don't know if I have that actually in my review. I should probably look. But I would like you to just have a general understanding of these hypersensitivities and just general vocabulary from here, like know what anaphylactic shock is, or, hives, allergic rhinitis, things like that. You don't have to tell me all the steps though, just some vocabulary out of it. Well, those are the main things from the immune system. I know I haven't covered everything in the 38 minutes here, but hopefully some of those tougher things are maybe clearer. So anything, okay, good. Anything else, like anything you want me to revisit that maybe I haven't done yet?
I have one, table 22.3, where they have the description of the type of this, like, for example, skin is stratified squamous. Do we have to memorize things like that?
Oh, yeah, no, no. That's huge.
Yeah. And also, could you tell us what kind of concepts we could focus on for the critical thinking type of questions? What kind of concepts we need to kind of think about?
Well, that's a good question. I don't know if you're going to have a lot of critical thinking stuff actually on this one, if I remember correctly, but as long as you are understanding the main concepts, like, how the cells are activated, how inflammation works, what it does to your body, for example, you would be able to answer application questions. But I don't think I have a lot of critical application questions on this one.
And then should we also have a good amount of ability to label because there are a lot of figures in this one with different specific types of cells? Should we know which is which?
I'm not gonna ask you like mast cell versus basophil versus neutrophil. I'm not gonna ask you that based on appearance. For the no, yeah, no, I'm not gonna make you tell me, is this a macrophage? So no.
But the process of inflammation, for example, could it be in like the last one, the heart was in a sequence?
Oh yeah. I kind of wish they had one like that for inflammation. I don't think they have one like that. But it could be as a sequence, like not labeling, it could be a sequence, yes. As like maybe like matching or putting them in order, it's just not going to be on a picture. But yeah,
Okay.
definitely know the steps, the 4 things, kind of the general steps of how the inflammation does happen.
Okay. And I'm guessing table 22.4, the chemicals of inflammation is an important one too, right?
20, hold on, table. Yes, where is it? No. No histamine, because I mentioned histamine and heparin both with mast cells and basophils. I'm not gonna make you match these things.
And leukotrienes?
No. Yeah. No, not of those. So you don't have to really focus on that table 22.4.
Okay.
It's a good reference table, like if you want to, remind yourself at some point later in your career, it's a good thing to refer to, but I'm not going to test you on that.
Yeah, okay. Thank you.
All right.
Seems like 22 is the more heavily focused on. Is it going to be mostly like 60 or 70% of the test?
Yeah, because it's it's much larger chapter too.
Yeah, okay.
All right, what else?
Oh yeah, what from the histology files do we need to know?
What do we have there? I think we have thymus. What do we have for? Well, we have thymus. I know that. Lymph node. Yeah, so this would be chapter 21. So we have a lymph node, you have thymus, spleen, and I don't have tonsils. So thymus, lymph node, spleen. I think that's it. Yeah, because there's nothing really with 22. All right, anything else? Okay, no. So the question is 3 levels of immunity: adaptive, innate, or immune. It's 3 lines of defense. The first 2 are within the innate. The last one is the adaptive. So you have first line of defense is preventing entry, and that's part of the innate. Second line of defense is internal defenses, and under that, you're going to see the cellular response, non-specific cellular response. You're going to see action of chemicals such as interferon or complement system, or physiologic processes, again, still underneath that second line of defense, like inflammation and fever. That's non-specific second line defense. And then third line of defense is the adaptive, the whole adaptive thing. So preventing entry is first, internal, non-specific internal defenses is the second, adaptive is the third. All right. Okay, if you come across anything, you have a question, you can text me or email when you think of something later on. And we have our exam is on Wednesday, due by Wednesday, right? 22-1, 22-1. 22-1, figure 22. Oh, to a table. Let me see. But yeah, that's a good summary. I mean, I wouldn't memorize the table. If you read that page, then that basically tells you what is summarized in the table. But just kind of be aware of what are different pathogens that can cause harm. Know that viruses are smaller than bacteria, much smaller. So I think, yeah.
So in terms of abnormalities, we should know things like allergies.
Or is that from the last part of 22? Is that?
Yeah. Yes.
Yeah. So there, yeah. Just know like briefly what each of those terms mean. I'm not gonna make you organize the phases in order though. Just want you to know, like, allergic rhinitis, hives, anaphylactic shock, know what that is. And okay. And those, yeah, I guess the three types of the hypersensitivities, acute would be like immediate, subacute is a little delayed, and then delayed hypersensitivity is later, like days. So, all right. Well, I mean, don't be afraid to send a question anytime. I'll try to get back to you as soon as I can. Again, texting is probably the fastest because I don't have my email open all the time. So email might be delayed a little bit, but reminder to record. Yes, it was, I swear I put it there and something happened. Because I put it already for all the exams. I don't know what happened to the last one because I did it that day. So I'm gonna double check that the reminder is posted in both of my sections on Connect and for all of the exams. Okay. No, somebody else did mention that it wasn't there, so it was probably my error. So I'm gonna double check that right now. Okay, but I'm gonna give you a verbal reminder. Don't forget to record before you start taking the exam. And then I'll double check it. All right, well, I'll see you all on Wednesday. Good luck. Don't forget the exam is due by 5 p.m., not 11:59 or any other time of the day, 5 p.m. on Wednesday. And then I'll see you at 5 on Wednesday. All right. I'll see you guys later. You're welcome. See ya.